Hi, my name is Lindsay Wythe. I'm a recent graduate from the University of Arkansas's Food Science Department, and my research is on the effects of a yeast fermentate prebiotic feed additive on the lactic acid producing bacteria in an in vitro microaerophilic cecal culture model. One essential method to safeguard the food supply is investigating new tools to improve food animal health and production. Developing this tool for poultry first requires understanding the dose response of the feed and feed additives, which can be linked to changes in the microbiota. Probiotin, a yeast fermentate prebiotic compound derived from brewer's yeast, has been used successfully in cattle management programs to improve gut health and feed efficiency. Because anecdotal evidence suggests probiotin may be useful as a poultry feed supplement, the following study was conducted to determine the effects of including probiotin in chicken feed on the cecal microbiota. We hypothesize that probiotin will have a positive effect on lactic acid producing bacteria in an in vitro chicken ceca model. 10 chicken ceca were collected from a commercial processing plant from broiler chickens. The ceca contents were squeezed out, homogenized, and 1 to 10 serial diluted. 1 milliliter of the final dilution was added to 300 milliliters of MHB. 20 milliliters was then transferred to 6 serum bottles per cecal dilution for an N of 10 and K of 6. The feed treatments used were the basal diet plus 0, 0.5, 1, 1.5, 2, or 3% weight per volume probiotin, with time collections at 0 and 24 hours post-culture initiation. Immediately following the addition of the ceca culture to the individual treatments, bottles were mixed, pressurized to the standard gut pressure using microaerophilic gases, and incubated at 37 degrees Celsius for 24 hours at 150 RPM. Ceca cultures were evaluated for lactic acid bacteria and total aerobic bacteria load changes over time. The CFU counts were log base 10 transformed, with data analyzed using two-way ANOVA, with the means separated using 2K's protected HSD, with a p-value of less than 0.05. The results of the current study demonstrated a significant interaction of treatment and time, as well as the main effect for lactic acid populations. For total aerobic bacteria, no difference was found between treatment groups at 0 hours, with a mean of 5.8 log base 10 CFUs per milliliter. At 24 hours, 3% probiotin had the least amount of lactic acid bacteria at 9.16 log base 10 CFUs per milliliter compared to other treatment groups. All groups demonstrated at least a 4 log base 10 CFUs per milliliter increase in lactic acid bacteria load between 0 and 24 hours. The 2% probiotin treatment group had significantly lower lactic acid producing bacteria loads than the 0% and 0.5% treatment groups, and was not different from the 1.1% group. The 0.5% and 1.5% groups were no different than the no treatment control. Based on the data presented, 3% probiotin demonstrates the greatest decrease in lactic acid bacteria load as compared to the other treatment groups, while 0.5% through 1.5% have the greatest increase in lactic acid bacteria populations. Moving forward, 0.5% through 1.5% may have the best results when included in a poultry diet. However, further studies evaluating the microbial ecology will need to be conducted to understand the effects the supplements in these populations have on the microbiome.